Hey everybody, Organized Biology here, and today we're talking about long-term potentiation. It's going to answer the question, how are memories stored? So what I've got here are two neurons. We're going to simplify this. This neuron obviously has a lot of stuff in it that's going to deal with storing memories, but this is going to send an action potential eventually, and we know action potential is just positive charges, right? So this guy's going to go from negative to positive eventually. And if we send that action potential, if we get this neuron positively charged, we're going to say a memory will be stored. Memory stored if action potential. Okay, so if we get this guy positive, we're going to store a memory. So the whole goal basically of this whole process is get this guy from negative charge to positive charge using a couple ions, calcium and sodium. We'll see how they kind of interplay. Now the second neuron over here, this presynaptic neuron, right? Presynaptic, postsynaptic. This guy is going to deal with actions regarding the information. Okay, what does that mean? Actions regarding the information. Well, let's say, for example, you are studying for a test, right? You want to learn about some sort of topic. So what you're going to do is you are going to begin looking at the diagrams. You're going to be rewriting your notes. You're going to be telling somebody about those things, right? All of these things will trigger this neuron, which is generally negatively charged like all neurons. And when you do these actions, it will make this neuron positively charged, therefore sending that action potential. Okay, so these guys, whenever you're doing the actions, send the action potential. So what happens next? Well, obviously we want to tell this neuron to get stimulated so we can store these information bits as memory. How do we do that? Let's get started. So the first thing that will happen is calcium will actually enter into this neuron. We've talked about this in previous videos where voltage-gated calcium channels are going to open up, therefore allowing calcium to flow high outside the cells into the cells. Okay, so calcium is now present inside of these cells. We know that calcium is a big mover in these neurons, so we're going to begin pushing these vesicles containing neurotransmitters out into this synapse. This neurotransmitter specifically is called glutamate. So I'm going to label this guy as glutamate. This guy's going to be a stimulatory neurotransmitter in this case, but it can also act as an inhibitory neurotransmitter other places. So we're going to release it into that synapse, okay? Just a little bit, maybe just a couple, okay? So you're starting to study, right? You're thinking about these things, and you begin releasing glutamate. Well, obviously, this glutamate can bind to some things, right? The first thing it's going to bind to are these things called AMPA receptor. They are ligand-gated what channels? Well, ligand-gated lock and key. So we're going to put the key, glutamate, into the lock, the ligand receptor. What are we going to open up? Well, these guys are red, and because they are going to be sodium channels. We know sodium likes to be high outside of cells, so therefore sodium will begin flowing into this cell. So we're just going to draw some sodium, and I'll draw some positive charges here. Great. Okay, so hey, is the goal reached? Well, the goal was take this guy from negative to positive, correct? So we're kind of bringing some positives in, which is fantastic, but usually this alone is not enough to actually stimulate an action potential. So this is not enough to stimulate action potentials. Now it can, but most of the time it doesn't. Now, let me give you an example of this, right? Let's say you're studying for, say, 2 minutes. 2 minutes, you're studying, you're releasing some glutamate, and then you go on a walk, and then you talk to your friends, and you come back to that same thing about a day later. Do you remember that information? Generally not. Generally not, because we didn't have enough action studying to cause a long-term positive charge in this neuron to get a memory stored, right? So the memory is not stored yet. In fact, that's actually called your working memory, where you can remember things for a few minutes, but you're not actually making this guy positive very often, okay? So we need to get even more information used and studied so that we can cause a long-term change so we can get this guy positive more. So let's see what happens next. Well, let's say you're going to continue to study. You're going to continue to study. And so you're releasing more and more and more glutamate in here. More and more glutamate will then trigger a different type of ligand channel. So we're going to come over here to something called an NMDA receptor channel. And as you can see with this guy, we've got a few players here. First off, it's not only a sodium channel, but it's a sodium and a what channel? Calcium channel, right? So we got a sodium and calcium channel. So therefore, whenever it opens up, we're going to allow both of these guys in. Well, how do we do that? How do we do that? Well, what's really fascinating here is as this neuron gets slightly positive, right? Slightly positive here, this cell is also very high in something called magnesium ions. We wrote magnesium ions over here. So we're going to have magnesium ions here that are what charge? Well, magnesium is actually a 2+ charge inside of itself. And so as this neuron gets slightly positive, this magnesium is like, whoa, whoa, whoa. Likes repel, correct? Positive charges don't like to hang out near other positive charges. So what's kind of interesting is this magnesium will begin to want to flow outward. And there's already a magnesium ion in this channel. So we're going to let that guy get kicked out because it's going to get pushed by that gradient. So now magnesium leaves, and guess what? If magnesium leaves, now we have an opening. Now we have an opening for both calcium ions to flow in as well as sodium ions to flow in. So as we're seeing here, we are getting a lot more positive charges into the cell, which is the goal, correct? So now we've got calcium and sodium inside of this neuron. And this is likely going to trigger an action potential, likely going to trigger an action potential. Why is that? Well, obviously we've got a lot of positive charges flowing in. Second thing is calcium here, so you see how calcium's coming in, is going to trigger what's called a secondary messenger response. This is a very complex process that happens in a variety of cells, but we're going to simplify it a little bit. So what is calcium going to bind to? Well, you see calcium comes over here and binds to a little protein here called calmodulin. And I always remember calmodulin, it's like calcium modulated. It's modulated by calcium. So when calcium comes in, we're actually going to kickstart a process. Well, what's the process? Well, calmodulin is bound to something called a protein kinase II. And this becomes activated when calcium binds calmodulin. Okay, so calcium binds protein calmodulin. That activates a protein kinase. So this is the protein kinase here. So if I were to separate these, we got calmodulin here. This is the protein kinase. Now, what the heck is a protein kinase? Well, lots of things to break down here. Well, kinase always refers to an enzyme, right? And kine always refers to adding a phosphate group to activate something. Adding a phosphate group to activate something. So you'll see kinases all over the place in different cell biology topics where it's going to add a phosphate to something and further activate it. So let's see what we're going to further activate with this protein kinase. Well, one of the things we're going to do is we're going to add one of these phosphate groups to this receptor channel. So now a phosphate group has been added to the receptor channel. Well, what does that do to the channel? Further activates it, right? So if we further activate this channel, what's going to continue happening? Well, more and more calcium is going to flow in, right? More and more sodium is going to flow in, right? So we're going to further stimulate this. You see how this is almost amplifying this effect? It's almost like a positive feedback loop, right? So the more calcium we get, even further more calcium we get, more calcium. Okay, so that's fantastic. Second thing we see is this protein kinase, along with the calcium itself, is going to come and stimulate the pushing out of these vesicles. So what these vesicles will do is they will get pushed to the outside. We know vesicles contains membrane, right? This is a cell membrane embedded with proteins. What are these proteins that you see? They are the AMPA channels, right? So now we are embedding more AMPA channels into the membrane. Well, what does it make more likely then? Well, it makes more likely that these are going to open, bind by, well, we got to draw the little receptor things right here. It's going to bind glutamate, right? If we bind glutamate here on both sides, Now what's going to happen? They're going to open up. What's sodium going to do? Further flow in, right? So now, oh my goodness, you see how just bringing in some calcium has now opened up this channel, pushed out these channels, and now we're allowing just a bunch of sodium, a bunch of calcium to flow into the cell. Is that a good thing? It certainly is, because all those are positive charges, thus making this neuron positively charged, thus sending the action potential. So we're just going to draw that now. We're going to draw very positive charges, and we're sending that action potential. What has now happened at this point is we have stored a short-term memory. So I'm going to draw right here. We have now stored a short-term memory. Now you may say, Mr. J, but all this work, all this studying just for a short-term memory? Yes. We'll get to long-term memory here in a second. But that is caused by an increase of these receptors at the membrane, right? Thus allowing more positive charges to flow in, thus allowing more action potentials, thus allowing more memories. Very cool. Now, if you have any questions so far, drop them in the comments below. Like and subscribe to this video. We're going to keep moving forward about long-term memory. Okay, so what's going to happen next? Well, we know that this protein kinase II can further cause other factors to be created. So one of those factors will be a nuclear effector. Okay, now, nuclear effector, my bad. Okay, so what is a nuclear effector? Well, it's any sort of molecule that can go into the nucleus and affect gene transcription, right? So what's going to happen here is we're going to have these little effectors come in and bind to specific nuclear receptors. That nuclear receptor in the DNA, this gene here is going to code for potentially a couple different things. We're going to say this gene, okay, so we'll just say gene 1, is going to code for other membrane channels. Other membrane channels. So what would that look like? Well, nuclear effector opens up this gene. What does that do? That gene will get read, transcribed onto, as we know, mRNA. We make an mRNA transcript for channels. That mRNA here is going to go to the ribosome, get translated, and now it's going to form, eventually, more channels. Those will get embedded in vesicles, right, just like earlier, and get further pushed out by calcium, thus producing more receptors on the membrane on the outside, thus making it more possible to send the action potential. So that's one amazing effect of this protein kinase, causing a downstream effect, making more channels and making it more likely for this guy to get sodium. Very cool. The second gene that we're talking about here, we'll just say gene 2, is going to code for different dendritic spine proteins. Dendritic spine proteins. Okay, what does that mean? Well, once again, gene is transcribed. We make that mRNA transcript. Cool. That goes to the ribosome. That will get read and produce a protein that is going to produce these proteins that are going to look like this. Okay, they're kind of like actin, they're microtubules with some actin in them potentially. And what these proteins are going to do, they're going to create what's called a dendritic spine. Well, what's a spine? Something that kind of pokes at you, right? Like the spines of a porcupine, or your spine has little outcroppings in it. And so what that is going to do, super fascinating, they will get pushed to the outside of the membrane and begin to push the membrane closer to that presynaptic neuron. So what will that look like? Well, we're pushing the membrane closer to this presynaptic neuron all over the place. So now the membrane's going to look more like this. You see what's happening here? So if we do that, why is this beneficial? Well, now this membrane has pushed closer to presynaptic neuron. In all reality, there's going to be other channels embedded here, just like earlier. And therefore, whenever you, say, go over your notes again, whenever you talk to somebody about the same information you were studying, that glutamate can just get to these receptors oh so much faster, get this guy positive from sodium coming in oh so much faster, and thus, boom, you can recall that memory very, very quickly. So all of these are mechanisms as to how we can get this neuron positively charged more often, recalling that memory, all from dedicated, focused work of focusing on specific information.